Hello students, today we are going to discuss in detail on replication error and the repair. Already we discussed about various type of mutation that is induced in the case of DNA, which includes transition, transversion, point mutation, insertion, deletion, etc. Further we discussed about how the DNA is broken by radiations. On exposure to radiation, harmful radiation, it results in single stranded break as well as double stranded break. And we discussed about induced mutation by various chemicals such as nitrous oxide, alkylating agents such as ethyl methyl sulfone and methyl methyl sulfone, MMS and EMS, etc. So when the mutations are very high in nature, these regions are called as hotspot, where several repeats of di, tri, tetranucleotides are present in the system, especially which is found in the microsatellites of the chromosome. Because it's very difficult to copy such repeats accurately and frequently, that undergo slippage. Either they increase in number of copies or decrease in the number of copies. For example, a dinucleotide repeat is Ca and a trinucleotide repeat is CGG and CAG. These repeats are seen in the case of adult muscular dystrophy as well as Fragile X syndrome and Huntington's disease. The trinucleotide repeat of CGG and CAG is slipped during the replication leads to increase in the frequency of this particular codon, which codes for glutamine, that resulting in the genetic disorder called as Huntington's disease. The CAG sequence codes for Huntington protein, which contains several stretch of glutamine residue. This polyglutamine patch interferes with the transcription in the gene of nervous tissue, which codes for the receptor. So it affects the brain of the person. Such replication errors escape proofreading, that is very difficult for us to remove from the DNA. There are certain mismatch bases arises during the time of replication by the DNA polymerase because it adds 1000 nucleotides per second. So enzyme, the DAM methylase, it adds methyl groups in GATC region, which is found in the case of both the strands of DNA. During the replication, the daughter DNA produced is semi-methylated, but newly synthesized strand is marked and it is recognized as a strand for repair. So this mismatched DNA is first recognized by the protein called as MUT-S, that is recognition complex. Then further MUT-L comes and joins to form the pre-incision complex. Pre-incision means it creates a cut, then followed by MUT-H, that is a incision complex. In the presence of ATP, the MUT-H comes and joins. Further, UVR-D, an helicase that binds with MUT-H, that is pre-incision complex, and it creates a single stranded break for the removal of the mismatch bases which is found. Once the gap is created, the gap is in turn filled by DNA polymerase 3 and ligated by DNA ligase. Here we can see the mutation repair mechanism in eukaryotes. which involves the protein MUT S, H and L. The eukaryotic system lacks methylation phenomena, and the repair machinery interacts with the sliding complex, that is pCNA, and recruited at the site of discontinuous DNA synthesis on the both leading as well as lagging strand. As we have seen already, that hereditary non-polyposis colorectal cancer, which is caused by the failure in the repair system of MSH2 and MLH when they are undergoing mutation. Here we can see the process of methylated and hemimethylated region. Now it is recognized by S, L and H, and how the DNA polymerase 3 is involved in the process. And the damage to DNA occurs on exposure to chemicals. Already we have seen that various type of mechanism that is involved: depurination, deamination, alkylation, oxidation, damage by radiation, base analogues, as well as intercalating agents we have seen. How this can be repaired? In the case of pyrimidine dimer formation, UVR photolyase, that is responsible for the separation of thymine dimer formation. The adjacent on exposure to ultraviolet radiation, adjacent thymine forms a thymine dimer or pyrimidine dimer. That time UVR photolyase comes and joins, and it breaks the bond between the adjacent pyrimidine dimer so that the thymine becomes normal during this process. When this enzyme DVR photolyase or DNA photolyase, when it is defective, that resulting in the clinical condition called as xeroderma pigmentosum, a clinically represented as a skin cancer. It is associated with various layers of the skin. When DNA is exposed to alkylating agents, the addition of methyl group takes place. That is, methylation takes place in the case of guanine as well as adenine. Once the methyl group is transferred to guanine, it becomes O6 methyl guanine. This methyl group is very bulky in nature, that is responsible for kink in the DNA or bend in the DNA, that is further removed by the action of the enzyme methyl transferase, so that the methyl group is eliminated and the DNA is restored to the normal condition by this process. And next one is excision repair, or we call it as base excision repair. When the DNA damage happens, the damaged portion of the DNA is flipped out of the helix. That time the DNA glycosylase enzyme has a catalytic cleft that glides over the double-stranded helix of the DNA. The flipped out base fits into the enzyme cleft and hydrolyzes the glycosidic bond. Thereby, the particular base is eliminated. That creates a AP site, that is apurinic or apyrimidic site, that is further removed by endonuclease. A gap is created that is in turn filled by DNA polymerase 1 and nick is sealed by DNA ligase. Normally, the base excision repair mechanism is seen in the case of deamination of cytosine to uracil, as well as it is also involved in recognizing oxoguanine creation, removal process, and it is also associated with deamination of 5-methyl cytosine to form thymine. Especially it is used in AT to GC mismatch repair mechanism. This enzyme is associated in the process. We call it as base excision repair mechanism. Another one is nucleotide excision repair, that is seen in the case of E. coli, in which the 4 proteins are involved: UVR A, B, C and D. The UVR A detects the distortion along with UVR B, which scans the helix. Then UVR A leaves, then at the same time UVR B melts the DNA, creating a
தெரியல
என்ன ஆன்சர் தெரியலன்றது தெரியலன்றது என்ன ஆன்சர்
தெரியலடி
என்ன தெரியல
அப்படின்னு அப்படியே பல் அடிக்கிற மாதிரி
அது ஒன்றும் இல்லை நீ இப்படியே படுத்துட்டு இருந்தால் நான் தலைகணையாவது எடுத்து வச்சு படுத்துட்டுருக்கேன். இப்படின்னா தலை ஒரு மாதிரி தான் ஆகும், வேறு ஒன்றும் கிடையாது. அது சாப்பிட்டு உடனே வந்து பிடிக்கிலேயே சாப்பிட்டு வந்து உடனே இது பண்ணிட்டேன். கொஞ்ச நாள் மாட்டிட்டு அது கொஞ்சம் 2 நடையாக நடந்துருக்கணும்.